Hello, I am Eduardo Vicente. I work at the Biogeco unit in RAE. And in this presentation, I will continue with the delta traits species distribution models applied to the early life stages of Abies alba, specifically applied to the process of germination and to fitness-related traits during the first months of life of the seedlings. The model, specifically the model that I'm going to present, is based on a translocation experiment using climatic chambers, and it uses the concept of climatic transfer distance, specifically temperature transfer distance. And we use temperature in this experiment because, as Marta told you before, the trees are very sensitive to temperature variations in these early life stages. The models that we use are generalized linear mixed effect models in which we regress the trait of interest against the climate of the population, the temperature transfer distance, and the interaction of the two. And these models also allow to add some covariables that may be also influencing this fitness-related trait. And the final goal, of course, is to obtain the distribution in the spatial distribution of the expression of this trait along the species range, and to make predictions of what may happen in future climate. To fit these models, we need enough biological and seed material from different provenances that gather enough climatic and genetic differentiation between them. And in this experiment, we use a significant number of seeds due to the low germination rate of the species. And we saw these seeds in 3 climatic chambers in which the temperature is set to mimic the temperatures experienced by the species in or during spring and during the growing season. And in this experiment, we split the seeds into batches. Half of the seeds were not stratified and sown directly into the climatic chambers, and the other half was cold stratified during 3 weeks and then sown in the climatic chamber for monitoring. And this is because normally this species requires between 3 and 6 weeks of cold stratification, although it has shallow dormancy, so it's still able to germinate without stratification. And in the climatic chambers, we make the phenotypic measurements of the functional traits of interest, mainly germination and its phenology, as well as emergence and its phenology, and several fitness-related traits of the seedlings exactly 2 months after leaf emergence. And these traits are, we focus on below ground and above ground traits. The models, the final form of the models is the following, although it's exactly the same as I presented before, with the nuance that as covariable, we added a seed mass and also the stratification treatment interacting with the climate of the population and transfer distance to account for potential differences in the dormancy depth between provenances. And to validate the models, we use the 64% of the data to calibrate them, and we kept 36% to validate the models. And in this presentation, I will just show quickly results of germination time and seedling height, seedling growth. Well, this is the germination time, which on one hand, we see that germination is hastened when the temperature transfer distance increases and also when the seeds are stratified, which is an expected result. But also we see here an interesting result showing that the seeds from provenances with colder springs, they are more sensitive to the stratification treatment. We see that the germination is quite delayed if they are not stratified, and it's quite hastened if there is a stratification treatment. And the results of seedling growth are also interesting. First of all, we see here that the maximum growth that we observe in our experiment happens at a transfer distance that is not at 0, but some degrees above 0. So that means that generally the species may enhance a little bit growth under a warming scenario. And here we see that this effect is more pronounced in the coldest provenance, or towards the coldest provenance, whereas the warmer ones, their optimum growth is observed close to the transfer distance of 0, which means that to a climate that is more similar than today's climate. And finally, these reaction norms, we can derive them into maps, into projections which may happen in the future, also with present conditions, and then make the difference, which is what I'm showing here. And here is a scenario of climate change in which we assume that the cold stratification is happening in both current and future conditions, and we make the difference between the two. And here in the left, we see the map for germination time, and the result here is saying that germination time may be hastened along all the species distribution range in these conditions. In the other hand, in the right hand, we see the predictions for seedling growth, or for the growth rate. And we see that the growth rate in this scenario is enhanced mainly in mountain ranges and in the north part of the distribution of the species, which correspond to the coldest areas of the species. On the contrary, the growth rate may be decreased in lower areas and plains surrounding mountain ranges, and especially in this area here of Poland, where I checked that the spring temperature is already a little bit warm for the performance of the species. And the second scenario that I present here is what may happen in assuming that the future conditions do not match the requirements for cold stratification. So this is the current conditions with the stratification versus the future without stratification. So for seedling growth, we have more or less exactly the same map as before, with the difference that now growth rate is generally decreased in all the distribution range of the species, but it remains similar in the mountain ranges and in the cold areas of the distribution of the species. For germination time, we see a much more nuanced map than before. And what we see here is that germination time is still hastened in lower areas and plains, and especially also in the south range of the species, whereas it may be delayed in the mountain ranges and in the cold areas of the species. And finally, as Marta said in her presentation, these models can be very useful and informative for assisted gene flow, and also for future generation programs that may take into account what may happen in future climate change conditions, but they must be also used with caution, because this is based in experimental conditions, which, for instance, do not take into account potential changes in soil moisture, for instance, because in our experiments, we only control the variable of temperature in the climatic chambers. And that's all for my part. Thank you very much for listening. And also thank you to the collaborators that made this experiment possible, and also to the funding institution.